Good afternoon. I'd like to welcome everyone. I'm Jay Magaziner. I'm the chair of the Department of Epidemiology and Public Health in the School of Medicine. And I'm honored to welcome you to the second annual Edson at Albuquerque Endowed Lecture in Translational Toxicology. This session, for those that are here, it's being, I guess, simulcast or telecast by Zoom to many people who are elsewhere around the globe. So welcome to you and you and you as well. After receiving his MD and PhD degrees in Brazil, Dr. Albuquerque received postdoctoral training at different institutions, including University of Illinois and the Karolinska Institute. In 1968, he was offered a faculty position at the University of Buffalo, where he established his first laboratory. He quickly rose through the ranks, becoming professor in 1972 and acting chair of pharmacology in 1973. Now, in 1974, as Dr. Albuquerque told it, and no one could tell it the way Dr. Albuquerque could tell it, a delegation was sent from the University of Maryland to recruit him to come to Maryland. Now, I don't know how accurate that is, but that's the story that I've been told, and I'm sticking to it until someone proves to me that it's different. In 1974, he became the chair of pharmacology, which he stayed in until 2010, when he decided to step down and move to a position where he could really develop his laboratory work even further. And we were fortunate that we were able to have him join us in the Department of Epidemiology and Public Health, where he established a new division of translational toxicology. During his long career, he published more than 400 articles and made major contributions to the fields of pharmacology and toxicology. He pioneered several electrophysiological approaches that enhanced the applicability of microelectrode techniques. His contributions span from studies of synaptic transmission at the neuromuscular junction to the central nervous system. In pharmacology, Edson's research provided the foundation for development of the nicotinic allosteric potentiating ligands, of which galantamine, a drug currently approved for the treatment of Alzheimer's disease, is a prototype. When he moved to toxicology and established the new division, his research led to better understanding of the long-lasting detrimental health effects of exposures to low levels of organophosphorus insecticides and nerve agents, and to the identification of potential new antidotes against their toxicity. His career was also marked by his mentorship of hundreds of young investigators in neuropharmacology and toxicology. He was an international leader. He worked with colleagues in Brazil and Europe and established an international training program in molecular pharmacology between the University of Maryland, School of Medicine, and the Federal University of Rio de Janeiro, which provided opportunities for graduate students from Brazil and the US to participate in exchange training. As a result of his internationally recognized contributions, Edson was the recipient of numerous awards. He was a member of the Brazilian Academy of Sciences, the Latin American Academy of Sciences, and Third World Academy of Sciences. Edson was an internationally acclaimed toxicologist, a preeminent scientist, a colleague, and a friend who is greatly missed by me and anyone who knew him. Not only was he a brilliant scientist, but he worked tirelessly, and I have to say his wit and amazing sense of humor made him a joy to work with. Edson cared deeply about the people who worked with him, and this lecture is a tribute to that. His work was groundbreaking, and it's a true privilege to carry on his legacy through this lecture. I'd like to thank Edson's family, friends, and colleagues for their generosity in making this annual lecture possible, and thank you all for being here to celebrate his legacy with us this afternoon. It's now my pleasure to introduce another colleague, Dr. Mordecai Blaustein, Professor Emeritus in the School of Medicine and former chair of the Department of Physiology in the School of Medicine. Notably, Dr. Blaustein was a close friend of Edson's. He also was mentor of today's special guest lecturer, Dr. Mark Nelson. And now, Dr. Blaustein will introduce Dr. Nelson.
Good afternoon, everyone. Good afternoon to the family who I assume are online, and of course to Edna, who asked me to come and participate. Now, as some of you know, when I wouldn't be standing here today if it were not for Edson, my friend and colleague. Now, I have to tell you that he chaired the Department of Physiology Chair Search Committee, and they did not send a delegation to get me, so when the dean first offered me the chair, I turned it down. But Edson was not to be, he didn't like to take no for an answer. And several months later, the dean called me back again. And so I visited, and Edson and I walked around Inner Harbor. That was just before the Inner Harbor and Federal Hill. We spent a couple of hours. It was before Harbor Place was built. But anyway, he was very convincing that I could help him to rejuvenate basic science here at Maryland. And the rest of that, of course, is history. Now, of course, Edson, as you've heard, was a really distinguished colleague and friend. Had the opportunity to collaborate with him on some work. He was a very impressive experimentalist. I marveled at the way he could poke neurons, and he was just superb at doing that. Now, you've also heard, or you can imagine, that Edson was somewhat bigger than life. And you've heard one story about it. Well, I can tell you that most of the stories that you hear are somewhat apocryphal. But I'll tell you one story just before I move on. I've got to tell you this. So shortly before I arrived, I actually started my position here. In the spring of 1979, I was visiting to get things straightened out. And Edson and I were scheduled to take a friend, Hugo Gonzalez, the late Hugo Gonzalez, out to dinner. He was looking at a position in the biophysics department. And I was in Hopkins Plaza, and of course that was the day before cell phone. So I went to the restaurant there, Café des Artistes, and they had an open phone booth, and I dialed the School of Medicine and told the operator that I didn't know his extension. Could she please connect me? And a very lovely female voice behind me said, "It's 76330. And raised my eyebrows, turned around, and my lower jaw dropped. Here was a stunning 30-some-year-old woman, and I really started to panic. I said, "My God, what am I getting into? Does everybody in Baltimore know Edson?" It turned out that she was assistant to Dean Bill Kennard. Bill was dean of the graduate school as well as the pharmacy school. And so Maybe he didn't know almost everybody, but I suspect he knew most people in Baltimore. Anyway, that's Edson, a real dear friend and colleague. Now, Mark Nelson. So you may have heard that Mark was my graduate student, my last student at Washington U, and passed. He was a very good student. He came here actually to finish his thesis, and then he spent a little time in John Lederer's lab before he moved to Constance, Germany, to work with my friend Peter Leuger in biophysics. And actually, Peter set him up with his protege, Ernst Bomberg, and Ernst became, who I had not met before, became a very good friend. In fact, I did a sabbatical with him. It was about a sabbatical year that I broke up into 15 years worth of sabbatical going back and forth. It was wonderful. Still a great friend. Anyway, while Mark was in Constance, he worked on, he put potassium channels into lipid bilayers, and that was the beginning of the study. Some of it was done here by Bruce Kruger with sodium channels. Mark did it with potassium channels and did some outstanding work. Then he came back here briefly before moving to Miami. And I don't remember when, but along the way, I offered him one piece of advice. When I was a student a trainee in Cambridge, a friend, Richard Adrian, who was a distinguished muscle physiologist, told me that his father, the Lord Adrian, who was a Nobel laureate for his work on sensory nerves, told Richard that if there's one thing he should not do, it's work on smooth muscle. Too hard. Well, I told Mark, that field is wide open. Nobody's studying it. You want to work on smooth, something to do if you want, if you have some time. Well, as you know, Mark worked briefly in Miami and then moved to Vermont in the Department of Pharmacology, moved up the ranks, became chairman of the department, and now is distinguished professor and chair of pharmacology. And along the way, he's done some really outstanding work on smooth muscle, not a variety of smooth muscles, but especially vascular smooth muscle. I'm not going to go through all of his honors. That would take me a long time. And I'm sure you don't want to hear from me about that. So, but he became a member of the National Academy. In fact, we celebrated with him about a year and a half ago. It was delayed a couple of years because of COVID. And of course, my wife Ellen and I got COVID while we were celebrating. Fortunately, it was quite mild. But anyway, we won't blame Mark for that. It was a great celebration. And anyway, Mark has trained a number of outstanding people as well. And we're very fortunate to have one of his trainees here in the department, Tom Longden, and I've joked with some of Tom's trainees that they're my scientific great-grandchildren. Anyway, so without further ado, since you really want to hear from Mark, he's going to tell us about electrocalcium coupling in brain capillaries, short and long-range control of cerebral blood flow.
All right, thank you, Marty.
Okay, great.
All right. Well, thanks. It's a great honor to be here, and to honor Edson and Morty is the greatest. I owe a lot to him. Lord Adrian also said, always put an equation in your paper too. And since we're telling a couple little stories, when Morty was looking for the chair job, we in the lab at Washington University knew exactly what he was doing at all times. He thought we didn't know anything. And so one day, Morty's son, Mark Blaustein, with a C, by the way, shows up with some female friend from high school. And he comes to see his father. And he says, Mark, where's Dad? I said, well, he's at the University of Michigan interviewing for a chair job. And he says, what? And the young woman looked at him, you don't know where your father is? So that's Then the second story, which I told Ed and others today, was after I went to Germany, I came back 6 months later to visit. I was walking down the road near Howard Hall, and Edson comes running at me. He goes, "Mark," and I go, "Edson." He says, "I want to hire you right now and give you a tenure track assistant professor job." I said, "What?" I said, he took, remember Morty took you up to his office, brought Morty in. I said, "I just started in Germany." I mean, like, I wasn't really prepared for that, but that was his enthusiasm. Of course, he got wise and probably didn't continue with me anymore on that one. But then I remember being a few times in his lab where he was so enthused about showing me what he was doing and kept talking and talking. I said, "Listen, I'm a busy person. I got to go and do something else." But his enthusiasm was really amazing. So I'm going to talk to you a little bit about, give you background on control of blood flow in the brain, what we're working on, the convergence of a number of studies, including with Tom Longden, and try to make it interesting. So Morty talked about the early days of vascular smooth muscle. Well, I was in neuroscience at WashU, and I don't know if you remember, Morty, Saturday seminar, 9 to 12 on Saturday morning for a 22-year-old. And the only thing they gave us was coffee and donuts. So I was shaking and sweating by the time the thing was over. But we never mentioned, well, only neurons, occasionally glia, which was kind of crap between the neurons, never mentioned blood vessels. Well, this shows that there are actually a lot of blood vessels in your brain, and they're in close proximity to all neurons. And this shows the surface peel arteries as they plunge into the brain, and they form this huge network of capillaries. And a little also introduction to part of the talk is that reduced blood flow in the brain is observed prior to overt clinical symptoms in diseases such as Alzheimer's, small vessel disease, which I'll talk about, and hypertension. And this just shows a little another movie, I hope I'm running, of a penetrating arterial. This little background. So these are some factoids. The brain represents 2% of the body mass, consumes a substantial fraction of the body energy. Neurons have limited energy reserves, so they depend on-time delivery of oxygen and glucose. And this is called functional hyperemia. Function of neurons, hyperemia blood, so increase in blood flow. So you can imagine you're sitting here semi-conscious, your eyes are open, your visual cortex is working, blood's flowing there. Then your eyes close, you pass out, and then you start twitching, your motor cortex works. This has to work seamlessly for decades in most people. And in competition with this, the other important feature of brain blood flow is autoregulation. And this is to maintain constant blood flow in the face of fluctuations in blood pressure. So they're almost competing activities. One is that the active neurons send a signal upstream to the arterioles to dilate, to deliver blood. At the same time, they want to maintain constant blood flow. And so it's sort of a kinetic issue, who wins first. This is a little bit of background. Oh, I hope I have the right talk here. Well, we'll find out. Anyway, this shows functional hyperemia in a mouse model. And you can see the neural activity goes up in the barrel cortex, wiggle the whiskers. Blood flow is delivered rapidly. This is described in the 1900s by Roy and Sherrington. The prize will go to anybody that can identify Roy or Sherrington. Sherrington won the Nobel Prize about 30 years. You should know, Tom. Okay, see? And this functional hyperemia is the basis of functional fMRI, which is used to map brain function. So a little bit about autoregulation. This is a remarkable feature. Over a wide range of blood pressure, shown here, between 70 and about 150 millimeters of mercury, blood flow remains constant. And we're still trying to figure out how this happens. But you can imagine, you get up, you pass out, whatever, and you change posture, you don't want blood flow to the brain to change dramatically. So it's an important feature. This also is compromised early on in disease. and as I just mentioned, functional hyperemia. This is to deliver blood locally and rapidly to places of active brain function. But keep in mind, the other thing, it's sort of like, we talk about climate change now, but in the early 1600s, the Dutch managed to turn Lower Manhattan into a sewer, because just like any other place, food comes in, fishing, but the waste has to go out. So there's a lot of metabolic waste. So probably an important feature of functional hyperemia is to get rid of metabolic waste. And you can think about it, the fastest way you can do it is through the circulatory system. So this shows ancient data, but makes a point. This is from the 90s. And so what we did was take a pressurized cerebral artery, increase the pressure from 60 to 100 millimeters of mercury, and the artery, and we have a calcium indicator in it. And the artery, you can see here, the calcium goes up, the artery constricts rather. In this case, it almost remains the same. And then we put a calcium channel blocker that prevents calcium from going into the smooth muscle cells, and the arteries dilate and calcium goes down. So this is a basic feature of, we think, is a major component of autoregulation. Pressure goes up, smooth muscle cells become more positive, the calcium entry goes up, and this is sensitive to one of the major classes of antihypertensives. One in particular, amlodipine is the one that's used most commonly, but there are a lot of different calcium channel antagonists of this class. So a simplified smooth muscle mechanism, mechanical stimulation, the membrane potential of the smooth muscle cell becomes more positive, calcium goes in, this causes contraction and vasoconstriction. So just sort of giving you a simplified overview. Now, in retrospect, everything becomes obvious. When this shows a study where the investigators, Anna DeVore and colleagues, stimulated whiskers and looked at the ascending vasodilation as it moves up from the inside the cortex to the surface. And they could measure the dilation as it ascends during stimulation, and they measured a velocity of about 2 millimeters a second. This doesn't sound like a lot if you're talking about stubbing your toe and getting a reaction in your brain at meters per second, but it's pretty fast. So this establishes functional hyperemia having to be fast. And then this also shows an archival plot of showing membrane potential of the smooth muscle cell versus diameter. And what you can clearly see at minus 40 millivolts, the arteries are constricted, and by simply making the membrane potential slightly more negative, we can get a complete dilation. So this is a powerful mechanism for controlling blood vessel diameter and blood flow. And there's a whole class of drugs that work simply by making the membrane potential of the smooth muscle cells more negative. And Tom works on some of those. All right, a little bit of factoids. This is a cast of a human brain. Your brain has about 500 miles of blood vessels. Some people have more, some people have less. Depends on where you are. If you go further north, you have more blood vessels than the south. No comment. And you have about 50 billion endothelial cells. And endothelial cells line all the arteries, and they make up the capillaries. So if you took your whole body, you have about 10 trillion endothelial cells that amount for several percent of the mass of your body. And most of the blood vessels are capillaries, which are made up of a single cell type called endothelial cell. They also form the blood-brain barrier that keeps stuff out of the brain, which is good and bad. If you think about it, well, you don't want stuff in your blood just going into the brain and affecting neurons. The flip side is you have brain cancer and you want that anti-cancer drug to get across the blood-brain barrier. Then you want to figure out ways to make the blood-brain barrier transiently leaky. The other important feature of these endothelial cells is they're electrically coupled by gap junctions. So if you look at this vascular cast there on the left, imagine those are electrical wires that go through your whole brain, sending electrical signals from cell to cell, which I'll show you some evidence for that. And the second thing is that all the capillaries are right next to neurons. So if you think of this thing, fast upward propagation from inside the brain after stimulation to the surface, and the fact that the capillaries are near all the neurons suggests, well, maybe the capillaries are the sensors. And when we first started this, and this is the halcyon days of Tom Longden back in Vermont. We were saying, well, I said, Tom, what about these? We're working on a grant. And he said, what about the capillaries? And he said, next thing you know, he shows me recordings from brain capillary endothelial cells. I said, I got to like this guy. I just suggest something. He often goes off and does it. Or to other people, I suggest stuff and they go off and don't do it or don't even pay attention. But if you think about it, in retrospect, how can you have a fast electrical signaling? It has to be retrograde because it has to start from within the cortex. And it has to be the capillaries because they're next to every neuron. So I'm going to provide some evidence about how this works. This is a basic system. So we suggest that active neurons, when they repolarize, they release potassium ions, and they can also be released from astrocytes. These potassium ions then go on, activate a particular ion channel, the inward rectifier it's called, that then turns it on, makes the membrane potential more negative. That electrical signal goes from to the next cell, to the next cell, to the upstream arterial to cause vasodilation. And so we set out to try to test this. I threw this in for any biophysicists just for fun. Since he's not here to defend himself, but one of our colleagues, Dick Chen, said after one meeting, he said, that was a great talk, Mark, but you should have been more didactic about how the system works. And I said, Dick, it was a 20-minute talk at 9 o'clock at night. Nobody wants to hear about this. So then he had me to NYU, and I said, I gave him 5 slides on how the inward rectifier worked. And I said, blame Dick. But basically, the idea, as you see in the cartoon, is that polyamines on the inside plug the pore. So when potassium goes up, it's like billiard balls and knocks the polyamines out of the pore, letting potassium to go through the pore. Because the polyamines are heavily positively charged, when you make the inside of the cell membrane more negative, it pulls the polyamines out. So on the left, what you can see is the increase in open activity with potassium. So if you go from 3 millimolar to 8, that's a 62-fold increase in its activity. But then that causes a hyperpolarization, a more negative membrane potential, and the membrane potential goes 30 millivolts negative. So you end up having a 1,000-fold increase in the activity of the channel. So it's a simple protein, but exquisitely tuned to make sure that electrical signals work. And then along the line, we were all talking about this, and Fabrice was in the lab, Fabrice Dabatron. And, I asked Fabrice, what do you do with he cannulates these 20 micron diameter arterioles from within the brain. And I said, what do you do with the capillaries? Oh, I just cut them off. So we'll just leave them on. And so he left them on. So this meant we could stimulate the ends of the capillary in green and look upstream at the arterial in an ex vivo prep to measure diameter, calcium, and other things. And as Tom remembers, we wanted to call the preparation La Fabrice, but Nature Neuroscience didn't go for that. I don't know why, but so this shows a cartoon of what we're doing. Picospritz, 10 millimolar potassium, and then we can use a blocker of this inward rectifier potassium channel barium ions. It eliminates the dilation to potassium, or if we knock the ion channel out of the endothelium alone, the propagation rate we can measure by putting in a microelectrode in, and it's precisely what Anna DeVore measured as the ascending dilation, about 1 to 2 millimeters a second. So let's see if this runs. Nah, damn. Right this way. Oh, codec unavailable. Well, anyway, you'll see the dilation to potassium. You're going. This shows it again. This time, we're measuring membrane potential with a microelectrode. The arterioles are are pressurized. And what you can see here in the bottom, you apply 10 millimolar potassium to the capillaries, and the smooth muscle cells hyperpolarize by about 30 millivolts. And the hyperpolarization starts within 200 milliseconds of applying potassium a millimeter away. And if you look up above, you can see the dilation to potassium and the reconstriction. And if we knock the ion channel out of the endothelium, only out of the endothelium, there is no dilation. So we come to this mechanism that capillaries are sensors of neural activity, and they sense changes in extracellular potassium to send a retrograde electrical signal that then goes to the smooth muscle cells, relaxes the arteries, increases blood flow. And more cartoons. Neural activity, potassium goes up, causes an ascending negative electrical signal that causes dilation to send the blood downstream. And so in these days, these heady days with Tom Longden and Fabrice, we got a little whimsical. So we called it the shadow nervous system because it was monitoring the activity of the central nervous system. And then I got even more out of control, and I gave this big talk for Paul Van Huda talk. And so I put Orson Welles and the shadow nose in there for anybody that was actually can remember that, but some people do. Okay. All right. So while we're doing this, I started a project with Ann Jutel, funded by the Leduc Foundation, on pathogenesis of small vessel disease of the brain, which, by the way, I really didn't know anything about at the time. And so we got the grant, and we started a great collaboration looking at changes in blood vessel blood flow properties in CADASIL. And CADASIL stands for cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. Rolls off the tongue. It's the most common monogenic cause of small vessel disease. There's about 1 in 50,000 people. You surely have people that are at this hospital. It's one of the leading causes of age-related cognitive decline. Also, other types of small vessel disease are responsible for spontaneous brain bleeds. And CADASIL patients, it's autosomal dominant and some mutation in the Notch3 gene. They know about it early on. This shows the gene. And there are modifications in the extracellular domain of the Notch3 gene. And so what we did, or I did, Ann did, or had done, we made mouse models with the human mutation. And they have all the same salient features as a human disease. And when they have deposits on the blood vessels, small blood vessels in the brain. And by the way, Notch3 is only expressed in smooth muscle cells and pericytes. And if Ann analyzes the proteins in those deposits and compares them to the proteins in the same deposits post-mortem from humans, they're the same proteins. They also have a deficit in functional hyperemia. We had a patient group part of our network in France, and they have deficits in functional hyperemia. So, and this sort of shows the basic feature of it. So one thing commonly done is called laser Doppler fluorometry. And the main sensory input for a rodent and some males, their whiskers, right, Tom? Okay. And so wiggle the whiskers and then measure blood flow in the part of the brain called the somatosensory cortex that responds to it. And what you can see in the middle or in the control is you can see an increase in blue, and that is the increase in blood flow to whisker stimulation. If we add barium ions at low concentrations that block the inward rectifier, we reduce this increase by about 70%. But note, in the CADASIL, the increase in blood flow is already reduced by 70%. And barium, which blocks the capillary-induced inward rectifier component, I know it's a mouthful, is completely gone. So it looks like this mechanism that I was showing you is completely crippled in CADASIL. And you think about it, serendipity, we were looking at the physiology for years about the inward rectifier, and we're doing small vessel disease in parallel, and I didn't think that they both would converge like that. So, so this is the mechanism, which I just showed you before. K goes up during neuronal activity. Barium blocks the pore. I don't show many current voltage relationships anymore because it puts people to sleep instantly, but this is actually a very characteristic inward rectifier potassium current on the left. And Osama did this work, and it's reduced by about 50% in CADASIL. So the channels aren't functioning. And I also should say, just since Morty talked about history, Sir Bernard Katz wrote a paper in 1949, published in the Journal of Physiology, France, of Paris, on anomalous rectification about this current. So it's been known for a while. So we're thinking about what's going on here. And there are a bunch of studies showing that this particular protein requires a phospholipid called PIP2 for activity. No PIP2, the channel's not active. It's a minor phospholipid in the inner leaflet of the cell membrane. It was first described by Don Hilgeman, structure which is shown here by Rod McKinnon. Word sits on the inner leaflet. The main thing to remember is absolutely essential. No PIP2, the channels can be there, but not function. And the bottom line is, when we looked at it, and we could restore normal channel function and normal physiology by giving PIP2 back. So the number of ion channels remained the same, but it was a PIP2. Now look at this. This is from a CADASIL mouse. Apply potassium to the capillary. This would normally cause a robust dilation. Do it right on the arterial, dilates. This is amazing. Fabrice actually did 6 experiments before he bothered showing it to me because he didn't want to get in trouble. So now we put it in the bath. 15 minutes, restored normal dilation to potassium. Bam. So it really looked like, and the single cell stuff works by Osama showed that the number of channels remained constant. It was about half of them had been inactivated by loss of PIP2. So this shows it again, but in vivo. So we used a trick. So in CADASIL, the increase in blood flow to whisker stimulation is down by 50%. And what we did was we put it IV PIP2. So it's an anion. It will go into the endothelial cell membrane, but it won't go into the brain. And we restored normal increases in blood flow shown in the middle. And it was the barium sensitive component. So we show that the sensor we think is this potassium channel that senses extracellular potassium, sends electrical signals upstream rapidly to dilate. I didn't show you, but also the Alzheimer's disease, small vessel disease. This is crippled, so the increase in blood flow is reduced. And add PIP2 back, and we restore the currents at the single cell level, restore dilations in the Fabrice prep, ex vivo, and we restore in vivo increases in blood flow, all in 15 minutes or so. So we're now trying to look for a mimetic that would do the same thing that somebody can take over time. And then we'll give it all away. Now, just a little bit, because I put calcium in the title. This is one of my spiritual leaders, John Cleese. A little bling. This is our new prep, just for fun, from Nick Klug. This is an on-phos retinal prep. And what you can see is on the left, there's a micro bead. It goes in the ophthalmic artery and goes through the entire circulatory system and comes out the vein, ophthalmic vein. So we can do a lot of things with this. And on the right is now a tagged red blood cell. You can see it ripping around through this. This is just for fun. So we originally had a hypothesis that there might be calcium signaling going on in the capillaries. And so well, actually Tom did it. So we had a mouse from Mike Cutler-Koff that we put the GCAMP8 into the endothelial cells. And I think it's one of the first experiments we did, Tom. He said, oh my God, there's all these calcium signals throughout, it's in vivo in the brain. Because we had this vision that the capillaries, there's these little tubes and red blood cells go through them. And now we have the view that their electrical signaling is going on all the time. And now there's calcium signals going on all the time. And some of them lasted as long as 60 seconds, and they never left one cell. And I'll show you an example of that. Of course, it took us 4 years to actually get the analysis program out of grant, but so here's Tom when he's young. So this is 4D imaging, and what you'll see, the red blood cells are in vivo, 425 by 425, rotate around, mask the bigger vessels. So now all you see is capillaries, and now the z-axis is time, 4 minutes. What you can see is this calcium signals going off. This is going up in time. And what you also can see is they're not moving side to side, which means they're not propagating from one cell to another. And you can see some are extremely bright and big. So the calcium output varies about a factor of 10,000. And if you do some calculations, you get about 5,000 events per second per microliter of cortex. And a lot of this was done by Amreen, who's going to join the NIH soon and granted the programming. And just to tantalize you, this was actually something that Science Advances asked us to do. They asked us, what is the relationship between electrical signaling and the calcium signaling? So we put barium on, which we blocked in with rectifier, and the calcium signaling went down by 80%. And I said, what? Then I thought, this is a summer, we're revising the paper. I thought, oh, great. It's like one of these things, everything you know is wrong. But I didn't want to, I want to keep the paper going. So then we came up with a hypothesis that all these electrical signals, hyperpolarizations, by activating potassium channels in the endothelium in vivo, increases the driving force for calcium entry, likely through TRPV4 channels. No matter, calcium is a positively charged divalent cation. The more negative the interior is, it wants to pull calcium in. So the idea is that the more hyperpolarizing signals we have, more calcium, that activates nitric oxide, which causes local vasodilation. So we have a whole study testing this, but I won't bother you with too much of it. This shows prevalence plots of the same thing. So this is over 4 minutes. This is, and you'll see the calcium signal. Low system resources may affect your audio quality. Well, I'm not stopping. I'm almost done anyway. This is on the right is barium. You can see very few events. And some statistics. I'll just show you one more. So then we knocked it out, knocked this ion channel out, only out of the capillary and out of the endothelial cells. And the same thing, a huge reduction in calcium signaling. So I went to, I mean, maybe you're familiar with some of this. I went to a Gordon conference on excitation and contraction coupling this summer, and they stuck me in a ryanine receptors section. And I said, well, I haven't. And then Don Burrs, who was organized, said, well, can you talk about our SPARC, is your SPARC work? And I said, and it's smooth muscle. I said, this paper was published in 1995. And I said to the audience, like, you know, okay, the Rolling Stones can go on tour and make money, but nobody wants to hear about my paper from 1995. There's no oldies tour for sciences, but unless there are some exceptions. Anyway, this seems to be really key. So this is the idea of electrocalcium coupling. So it's about 400 microns in distance between the penetrating arteriole and the ascending venule. And if we have signals going at 2 millimeters a second, they start, and we picospritz, and they basically propagate, and they're out of view. But we see is an increase in open probability bidirectionally. So there doesn't seem to be any bias of one direction versus another, which I don't understand at the moment. Okay. So you can imagine as these neurons are active, they're hyperpolarized in the endothelium. These waves of electrical signaling, negative electrical signaling, are coursing through it, and then calcium entry is increasing to increase the probability of calcium signals. I didn't do that. And so you can imagine it's almost like dropping embers along the way into the endothelial cell, and they're activating an ion channel called IP3 receptors. So we did this also in mouse models that have compromised electrical signaling, and this just shows one example where there are almost no events in this Alzheimer's mouse model. the calcium signaling is lost, and so is the electrical signaling. Again, coming to the question, well, what happens if this goes on for, years? So this is our electrical calcium coupling. When I was in this session, it was funny. I said, okay, so the heart beats, excitation, calcium goes in, activates ryanine receptors, calcium transient, contraction. I said, I'm going to talk about EC coupling, but there's no voltage bending calcium channels, there are no ryanine receptors, and the voltage goes the other way. And I said, so this is basically the idea is that the calcium signals are generated by GQPCRs activating IP3 receptors, increasing calcium, nitric oxide, and then the hyperpolarization elevates calcium entry, which then increases the probability of all this happening. However, at the moment, I don't really, it's sort of the basic question, why is this happening? And why this higher order sort of organization? So I'll throw a few questions out, what happens if we can intervene, restore normal blood flow early on in small vessel disease? Are the paths continuous along the vascular tree? What happens if signals hit at junctions? What's the mechanism underlying calcium signal propagation, which I think we know. How's the balance determined? The signals seem to be bidirectional, but we really need to have a voltage indicator. And then how's the brain sort of organize competition for resource utilization? Because it's a finite, the brain is a finite volume, and you have all these different blood flow going on all the time. And what determines the strength of the signal is it plastic, which was Tom's looking at now. So take-home points: concept, rapidly spreading membrane hyperpolarization increases calcium entry through resident calcium permeable channels, which in turn increases the probability of calcium release, which increases calcium. So you can view the capillaries like electrical wires passing negative hyperpolarizing waves bidirectionally through an entire capillary network to ignite local calcium events to optimize blood flow and waste removal. So funding organizations, pretty much everybody. This is Messiah did a lot of the work. Bruno, Grant's done all the programming. Tom's actually been putting microelectrodes in pericytes and endothelial cells. This was COVID lab meeting at my house outdoors, since we didn't want to go anywhere. Some more pictures. A little bit about Vermont. We have a new research building. It's in our parking lot. This is Lake Champlain. Massachusetts is south. We seldom go there. It's too conservative. We'll go up to Quebec. And New York State, that's Plattsburgh. Did you ever go to Plattsburgh, Tom? And I want to thank everybody. This is a picture that Anne Reen took during leaf season last year.